Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Biomolecules. So in the last lecture, we studied about the lipids and we were seeing about the classification of lipids. Right? In the classification, we saw that lipids are basically of three types: simple lipids, conjugated or compound lipids, and derived lipids. Then we saw what are simple lipids and conjugated lipids. In the conjugated lipids, the category that we studied was phospholipid. The phospholipids are the major components of the cell wall, cell membrane, right? Now, that was the one example of the conjugated lipid. The second example of the conjugated lipid. So, the conjugated lipid, the second example is lipoproteins. Okay. Now, what are lipoproteins? So, as the name suggests, these are the lipids that are conjugated with proteins. Okay. So, lipoproteins are the lipids conjugated. These are the lipids conjugated with proteins. Okay. So, they have a lipid part and a protein part. Now, what is the function of this lipoproteins? So, the function of the lipoproteins is transportation. So, these lipoproteins helps in the transportation of the lipids. Okay. Now, these lipoproteins are of two types. The first one is the low density lipoprotein and the second one is the high density lipoproteins. So, what are low density lipoproteins? So, this lipoproteins, they transport. So, these lipoproteins transport cholesterol. So, both of these lipoproteins, they transport cholesterol, but where do they transport this cholesterol makes a difference. Okay. So the low density lipoproteins, what they do, they transport cholesterol. They transport cholesterol from liver. They transport cholesterol from liver to tissue. Okay. They transport cholesterol from liver to tissue. And how do they transport this? They transport it via blood. Okay. So the most of the transportation in our body takes place via blood. So these low density lipoproteins, they take a cholesterol from liver and then they transport it to the tissue. But these cholesterols that they transport to the tissue, these cholesterols are the bad cholesterols. Okay. So the cholesterol that they transport, these are the bad cholesterols because these cholesterols then go and deposit in the tissue and hence they increase the risk of cardiovascular disease. Okay. So these cholesterols, they increase the risk of cardiovascular disease. Okay. They increase the risk of cardiovascular diseases. Hence, in our diet, it's not good to take low density lipoproteins because these proteins, what they will do, they will accumulate the cholesterol, bad cholesterol into the tissue. That will increase the risk of cardiovascular disease such as heart attack. Okay. Now, the second category is high density lipoproteins. So these high density lipoproteins, what they do? So they transport, these lipoproteins transport cholesterol from blood to liver. Okay. From blood to liver. And what does the liver do? Liver metabolize this cholesterol. Okay. And hence these cholesterols are what? These are good cholesterols. Okay. These are good cholesterols. Hence, so based on this, what we can say, hence it is recommended to take high density lipoprotein in your diet, not the low density, because this low density will increase the bad cholesterol in our body, right? And that will lead to various diseases. Okay. So this is about the conjugated lipid. So in the conjugated lipid, we studied about the phospholipids, and then the second is the lipoproteins. Now, the third category or third type of lipid is the derived lipid. So what is derived lipid actually means? So derived means that it has modifications. Okay, it has modification of fatty acid. So derived lipid, in derived lipid, it has modification. It has modification of fatty acid. Okay, it has modification of fatty acids. Okay. Now, what are the examples? So the example, one of the example of derived lipid is esterols. Okay. One of the example of derived lipid is esterols. Now, what are sterols? Sterols are, these are what you in a ring structure. If you look at the structure of the sterol, these are ring or cyclic structures. So it consists of ring or cyclic structures. And the common sterol that is present in animals. Okay. So the common sterol present in animals is the common sterol present in animal is cholesterol. So the cholesterol that I was talking about in the previous slide. So this is a modified or derived lipid. Okay. And it is commonly present in the animals. Okay. Now, this cholesterol is not always bad. Okay. This is not always bad. Yeah. So this cholesterol is not always bad. If we talk about the structure of the cholesterol, so this is how it looks like. Okay. So this is what? This is cholesterol. This is cholesterol. So the cholesterol looks like this. And this cholesterol has not always a bad role, but it has various important roles, such as it produces hormones. So cholesterol, what it does, it is also part of cell wall. So you can write part of cell membrane. Sorry, part of cell membrane. First of all, it is part of cell membrane. Second, it produces hormones. And what hormones it produces? It produces the hormones known as steroids. The hormones that it produces are steroids. Examples are estrogen, and progesterone. Okay. So these are the certain hormones that are present in the that are produced by the cholesterols. Okay. Now the second type of derived lipid is prostaglandin. Okay. The second type of derived lipid is prostaglandins. Okay. So what are prostaglandins? So these are also a kind of derived lipid that is produced by the modification of. These are produced by the modifications. These are produced by the modifications of arachidonic acid. Arachidonic acid. So in the previous class, we saw what is arachidonic acid. So it is unsaturated acid, fatty acid that is of 20 carbon. 20 carbon, and it falls into the category of PUFA, polyunsaturated fatty acid, because it has 4 double bonds. It has 4 double bonds, right? So prostaglandin is a modification of arachidonic acid, and this prostaglandin, it is responsible for pain and muscle contraction during baby delivery. When the baby is delivered, so during that time, it is responsible for the pain and muscle contraction during delivery, right? So this is about the derived lipids. So with this, we have completed the types of lipid or the classification of the lipids on the basis of, you can say, their structure or the basis of their components, right? Now, there is one more classification of the lipids based on the. There is one more classification of the lipid. It is based on their production in the body. So this classification is based on their production in the body. So if you remember, we had also categorized amino acids in 2 categories, that is essential and non-essential amino acids, based on the ability of the body to produce them. So if body can produce it, then it is non-essential amino acid. But if we require it from outside, then it falls into the essential category because it is essential to have that thing or that amino acid in our diet. Similarly, the lipids are also classified on the basis of their production in the body in two categories, that is essential lipids and non-essential lipids. Now, what are essential lipids? These are the, like I said, lipids that cannot be synthesized. Cannot be synthesized in the body. It cannot be synthesized in the body, but are important. Right? So these are essential amino acids. And what are the examples of essential amino acids? Unsaturated lipids. Sorry, essential lipids. These are unsaturated lipids. Okay, like unsaturated lipids that I told is OLA, right? Oleic acid, linoleic acid, and arachidonic acid. So these are the unsaturated lipids that are important for our body but cannot be synthesized in the body, and hence it is required to have in the diet. Okay. Now, what are non-essential amino acids? So these are the amino acids that are synthesized in the body. So what are the essential lipids? These are the lipids that are synthesized in the body and are important for normal health and growth. Are important for normal health and growth, right? So this line is important because there are many lipids, right? Those are not healthy, right? So it does not mean that it will fall under essential or non-essential category. It won't if it's not healthy. So it should be healthy. And if it is synthesized in the body, then it is non-essential. And if it is not synthesized, then it is essential, right? And the examples are saturated fatty acid. Saturated fatty acid. Okay. Okay. So this is about the classification of lipids. So we saw we classify the lipids on two bases. One, on the basis of their structure of their component, that is simple, conjugated, and derived. And the second, on the basis of their production in the body. So with this, we have completed this another biomolecule, that is lipid. Now we are left with the last biomolecule, that is called a nucleic acid. Okay. So now we'll study about the nucleic acid. So the first question is, what is nucleic acid? So nucleic acid, it comprises DNA and RNA. Okay. So it comprises DNA and RNA. Now, what is DNA and what is RNA? So DNA stands for deoxyribonucleic acid. Okay, deoxyribonucleic acid. Okay, and this DNA in most of the organisms, except for few viruses, DNA is considered as genetic material. Okay, so it stands for deoxyribonucleic acid, and it is considered as genetic material. It is considered as genetic material in most of the organisms. Or you can also say all the organisms, because viruses, we do not consider them as a living organism, right? So it is a genetic material in, you can say, most or all of the organisms, if you're not considering virus as an organism, right? Now, the second type of nucleic acid or second component is RNA. Now, RNA stands for ribonucleic acid. It stands for ribonucleic acid. Now, what is ribonucleic acid? So, the ribonucleic acid, it is also considered as genetic material in some viruses, not all the viruses, but genetic material in some viruses. Okay. So it is considered genetic material in some viruses. And RNA is of 3 types. Okay. So the types of RNA is of 3 types. Okay. So the first type is known as mRNA. Now, what is mRNA? mRNA is messenger RNA. It is called as messenger RNA. And what it does, so what happens that we know that DNA is converted into RNA and RNA is converted into protein, right? This is what we have seen previously. And if not, we will see in detail in upcoming classes. Okay. So what happens? This is your cell. You have DNA. If suppose this is a eukaryotic cell, you have DNA. And then this DNA is converted into RNA. Okay. This is converted into RNA. And then this RNA comes into cytoplasm. So the DNA that is converted into RNA is this mRNA. Okay. So this mRNA, what it does? So it carries information. It carries information from DNA. Okay. So the DNA is getting converted into RNA, and this RNA that we are saying, not getting converted, you can say that DNA is encoding the RNA. Okay. So this RNA, it carries the information from the DNA. Okay. And this RNA is the messenger RNA. Now the second type of RNA is rRNA, and it is also known as, or it stands for ribosomal RNA. It stands for ribosomal RNA. And what it does, so ribosomal RNA, it is a part of ribosome. And what is ribosome? Ribosome is, we know, it is protein synthesis machinery or organelle. Protein synthesis organelle, right? Now, the third type of RNA is called as tRNA. So there are multiple other types of RNA, but these are 3 major types of RNA that we should know for now. So tRNA stands for transfer RNA. And this transfer RNA, for now, you just remember that it carries amino acid. Okay, it carries amino acids for protein synthesis. Okay, it carries amino acid for protein synthesis. Okay, so this is the type of RNAs that we know. So the nucleic acid, what we saw, that it comprises of DNA and RNA, and then what is DNA and what is RNA. Now, if we talk about the location of this DNA and RNA, so in the case of eukaryotic cell, eukaryotes, so the typical diagram is this only. So we have nucleus. So the DNA is present in the nucleus. DNA is in the nucleus. But the RNA, the messenger RNA is encoded in the nucleus, but it then comes outside in the cytoplasm, right? So you can write the RNA is present both in the nucleus and cytoplasm. Or if in the option only cytoplasm is given, then you can also opt for the cytoplasm. Okay. Now, in prokaryotes. So in prokaryotes, what happens? There is no well-defined organelle, right? Membrane-bound organelle. So everything in the prokaryote is present in the cytoplasm. Okay. So in prokaryote, DNA and RNA both are present in cytoplasm. Okay. So this is the location of nucleic acids in the case of eukaryotes and prokaryotes. Now, let's move forward and see the structure of the nucleic acid. So, if you look at the in NCERT, it is mentioned that nucleic acid, it is made up of heteropolymer of nucleotide. So, nucleic acid, it is made up of heteropolymers. It is made up of heteropolymer of nucleotides. Heteropolymer of nucleotides. Now, it has 2 terms. The first thing is written as heteropolymer, and the second is written as nucleotides. So what do we mean by heteropolymers? So till now we have developed this understanding that hetero means it has different monomeric units, right? So the nucleic acid do not have only one type of monomeric unit. It has different monomeric unit. So heteropolymer is different monomeric units. And we will see what are these monomeric units for now. So it is made up of different type of monomeric unit. The second term I'm talking about is nucleotides. Now, what is nucleotide? So before that, so since we are writing it that it is made up of heteropolymer of nucleotides, we can also say that nucleic acid is polynucleotide chain, right? Because it has multiple nucleotides. So nucleic acid is polynucleotide chain. So it is chain of multiple nucleotides. Okay. Now let's see what is nucleotide. So the nucleotide, the term nucleotide, it is made up of three components. So it comprises three components. Okay. The first component is sugar. So in the carbohydrate, we have understood what is sugar, right? So it consists of sugar. Second, it consists of nitrogenous base. It also consists of nitrogenous base. And then third, it consists of phosphate. It consists of phosphate group. Okay. So it has three components. The nucleotide has three components: sugar, nitrogenous base, and phosphate group. Okay. One more thing, this sugar and nitrogenous base, they together are known by a different term, and that is known as nucleoside. So the sugar and the nitrogenous base, they together are known by the term nucleoside. Okay. So we can also write what is nucleotide. So the first thing, the nucleotide components, you can say sugar, nitrogenous base, and phosphate group. But you can also say that nucleotide comprises of nucleoside plus phosphate group, right? Because nucleoside is what? Sugar plus nitrogenous base. So it comprises of nucleoside plus phosphate group. Okay. Okay. So this is about the structure of nucleic acid. Now let's study these components of the nucleotide one by one. So the first component is sugar. The first component is sugar. So in the case of nucleic acid, in nucleotide, what kind of sugar is present? Because we know that there are different types of sugar. So in nucleotide, the 5-carbon sugar is present. Okay, 5-carbon, or you can say the appropriate term will be nucleic acid. Okay, so in nucleic acid, what happens? In nucleic acid, 5-carbon sugar is present. Okay, the sugar that is present is 5-carbon. And what is this sugar? So there are two types of sugar. Both are 5-carbon. The first is ribose sugar. And this ribose sugar, it is present in RNA. Okay, this is present in RNA. So we saw nucleic acid is of two types, DNA and RNA. So the ribose sugar is present in RNA. Okay, now the second sugar is what? The second sugar is deoxyribose. It is called as deoxyribose. And this is the deoxyribose sugar. It is present in DNA. Okay. Now, if you look at their structure, what difference can we see in the case of ribose and deoxyribose? The difference that we can see is clearly marked here. And the difference is this 2' OH. So 2' is at carbon position 2 of this ring. Okay. So what we are seeing, so we can write difference between ribose and deoxyribose is difference between ribose and deoxyribose is, what is difference? That difference is at second carbon of sugar, right? At second carbon of sugar. What do we have? In the case of ribose, In the case of ribose, what do we have? We have OH. OH at second carbon, right? And in the case of DNA, or in the case of, you can say, deoxyribose, what do we have? We have H at 2' carbon, or at second carbon of the sugar, right? So this is the difference between the deoxyribose and ribose sugar, and that is ribose is present in the RNA, while the deoxyribose is present in the DNA. Okay, and hence their name comes from this only, because we know DNA stands for deoxyribose, deoxyribonucleic acid, right? So because of this, sugar is known as deoxyribose. It is called as deoxyribonucleic acid, while the RNA, the sugar is ribose, hence it is called as ribonucleic acid. Okay. Okay. So we have studied about the sugar, and what is important, that the sugar, it has 5 carbon sugar. Okay. Now, the second component is what? Nitrogenous base. The second component that is present in the nucleotide or nucleic acid is nitrogenous base. Now, what is nitrogenous base? Nitrogenous base is, it is in a heterocyclic ring. It is heterocyclic ring. Heterocyclic means that it has more than one type of atoms. Okay. Homocyclic means only one type of atom. It is made up of element. If it is made up of carbon, then the whole ring will consist of carbon. Heterocyclic means it has more than one type of element present in that ring. Okay. So it is heterocyclic ring with nitrogen. So it means it has this cycle has both nitrogen and carbon. Okay. And hence it is also known as nitrogenous base. Okay. Now, if we talk about the nature of this nitrogenous base, these are hydrophobic in nature. The nitrogenous bases are hydrophobic in nature. Just like in the case of lipids, we saw that the head portion is hydrophobic in nature. Similarly, these are hydrophobic in nature. Okay. So the tail portion is hydrophobic in nature, right? So this nitrogenous bases are hydrophobic in nature. Okay. Now, now we talk about this nitrogenous base. So nitrogenous base types. So the nitrogenous base is of two types. Okay. The nitrogenous base is of two types. And these types, let's divide from here. Okay. So these are the two types. So the first type is purine. So the structure that we are seeing here, or I can also draw a general structure that will look something like this. It will look, okay, and it has nitrogen, 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 and nitrogen. So this is the general structure of purine. Okay, so this is the general structure of purine. So what do we have in the purine? We can see it has double ring. Purine has double ring. And if I talk about the size, it is larger in size. Larger in size. Okay. It is larger in size. Now, if we see the numbering, so in the case of purine, the numbering starts from this nitrogen. This is 1, this is second position, this is third, this is 4th, 5th, 6th, 7th, 8th, and 9th. Okay. So in this, what we can see, we have 4 nitrogens. So in purine, we have 4 nitrogens. Okay. And this purine is further of two types. It is further of two types, first and second. The first here we are seeing is the structure of adenine. The first here is the structure of adenine, and the second is guanine. Okay. So the only difference what we are seeing is that 6th position and 2nd position compared to adenine and guanine. So in adenine, we have NS2 at the 6th position, while in the case of guanine, we have O, double bond O. Okay, we have amine group, and we have, you can say, ketone group in the case of this guanine, right? And then we have one difference at the 2nd position, right? So this is, both of these are purine. So purine is of two types, adenine and guanine. Now, the second category is called as pyrimidine. The second is called as pyrimidine. And if I draw the general structure of pyrimidine, it will be like this. Okay. It will have N here and N here. And then double bond, double bond. So this is the general structure of pyrimidine. Okay. Now, the pyrimidine, we can say it is single ring. So it is single ring. And how many nitrogens do it have? It have 2 nitrogens. It have 2 nitrogens, right? And it is comparatively smaller in size as compared to purine. It is smaller in size. Now, the pyrimidine is of 3 types. Okay. So the first pyrimidine that we are looking is thymine. The second is cytosine. And the third is uracil. So this is U. The third is uracil. Also, this adenine is represented by A, guanine is represented by G, thymine is by T, cytosine C, and uracil U. Okay. So what we are seeing? So we have nitrogenous base of 2 types. The first one is purine. The second is pyrimidine. Again, this purine is of 2 types. That is adenine and guanine. And the pyrimidine is of 3 types. The first one is thymine. Second one is cytosine. And third one is uracil. Now, how numbering is done in the case of pyrimidine? So in the case of pyrimidine, the numbering starts from here. Remember, in the case of purine, the numbering starts from here. But in the case of pyrimidine, the numbering starts from here. And then in anti-clockwise direction, 2, 3, 4, 5, and 6. So it has 6 members. It is 6 members then, right? While in the case of purine, it is 9 members. Okay. So this is about the nitrogenous base. Now, one more important thing, that in the case of RNA, in RNA, what do we have? We have adenine, we have guanine, we have cytosine, and you have uracil. We do not have thymine in the case of RNA. Similarly, in the case of DNA, we have adenine, we have guanine, we have thymine, and we have cytosine. We do not have uracil in the case of DNA. Okay. So this is the composition of DNA and RNA. Now, okay. So this is what we studied. We studied about sugar. We studied about the nitrogenous base. Now, before studying about the nucleoside, the structure of nucleoside, let's study about the phosphate group. Because what we said, that we have in our nucleic acid, we have 3 components. The first one is the sugar, the second is nitrogenous base, and the third is phosphate, right? So what is phosphate group? So the phosphate group in nucleotide is derived from phosphoric acid. So it is derived from phosphoric acid. And what is the structure of phosphoric acid? It is OH. So the phosphate group is derived from the phosphoric acid. Okay, this is you should remember. Now, let's move to the nucleoside. So what do we have in the nucleoside? So we know that nucleoside, it comprises of what? Sugar plus nitrogenous base, right? Sugar plus nitrogenous base. Now, let's draw sugar. So what we have? We have sugar. And we know what is that sugar? That is ribose sugar or deoxyribose sugar. But both are 5 carbon, right? Now, in this sugar, the ring structure, we have CS2OH here. And this is what? This is 5th position. This is 4th. This is 3rd, 2nd, and 1st position, right? We have 3rd position, we have OH. And this 2nd position, based on are we drawing deoxyribose or ribose, it will vary, right? Now, at the 1st position, we have what? OH. We have OH, right? So this is what? This is a ribose. You can say ribose. You can draw ribose by adding OH here, right? So this is a ribose sugar for now. Now, let's draw nitrogenous base, because what we are saying, nucleoside is the sugar plus nitrogenous base. So nitrogenous base, suppose I'm drawing purine for now. Purine is what? Double member ring. So this is my purine. Okay. This is my purine. We have N here. Rest of the structure I'm not drawing. So this is the 9th position, right? And this is N. This is the first position. And this N has H here. This N has H here. Now, this H and this OH, it will form a bond. And this will cause what? Release of water. It will cause release of water. And the nucleoside that will form will look like this. Will look like this. So here, suppose this nucleotide is what? You can say adenine. This is adenine. So I'm not drawing the whole structure. So the nucleotide here will be attached and this will be the adenine. Okay. And here you have rest of this thing will not change. So it will have CS2OH, it will have OH, it will have OH. So what we saw, and this bond, the bond that will be formed, this bond is known as glycosidic bond. Okay. This bond is known as glycosidic bond. So what we have seen here, we have seen that here it was purine, right? So in the case of purine, so the glycosidic bond is forming between which elements or which elements? So if we have purine, right? In the case of purine, what do we have? The bond is forming between carbon 1 of sugar. It is forming between carbon 1 of sugar and nitrogen 9. Nitrogen 9 of purine, right? So even if it has guanine, the bond will be formed between carbon 1 of the sugar and nitrogen 9 of the purine. In the case of pyrimidine, this will change. In the case of pyrimidine, what will happen? The bond is formed between carbon 1 of sugar and nitrogen 1 of pyrimidine. Okay. Nitrogen 1 of pyrimidine. Okay. So if we have pyrimidine, then this, because we have only one ring, right? In the case of pyrimidine, if we look at the structure, so the bond formation will take between this will involve in the bond formation. And this is what? Nitrogen 1. Okay. So the glycosidic bond is formed between the carbon 1 of the sugar or either nitrogen 9 if it's purine or nitrogen 1 if it is pyrimidine. Okay. And this structure is what? This is nucleoside. This structure is nucleoside. Okay. Now we have understood nucleoside. Now let's move to the nucleotide. So what do we have in the nucleotide? In the nucleotide, what we say? We say that nucleotide has nucleoside plus phosphate. Nucleoside plus phosphate. Right. It has nucleoside plus phosphate. Now, nucleoside structure we have just drawn. So we know the structure of nucleoside. The structure of nucleoside is like this. And here, suppose again, I'm taking adenine example. Now, in the nucleotide, and then you have here what? You have CS2OH, right? This is the fifth carbon that is consists of, this is the fifth carbon that is CS2OH. Now, we have to add phosphate group. Now, where we can add the phosphate group? This numbering is 1, 2, 3, 4, and this is 5. The phosphate group, it comes from phosphoric acid, like I said. So it will have what? It will have P, O, OH, OH, and OH. This is a phosphoric acid, right? This is a phosphoric acid. And this is what? This is a nucleoside. This is a nucleoside. Now the bond formation will take place between this H and this. Okay, this will release water, and the nucleotide that will form will be like this. Will be like, here you have your group. This is the glycosidic bond that we know. This is the glycosidic bond that we know. Right. And here we have CS2, O, P, O, OH, and OH. So this is what? This is your nucleotide. This structure, sorry. Okay, so this structure that has formed, it is what? It is, this structure is your nucleotide. Okay, now what is this bond? The bond that is formed, this CS2, this bond, this bond is called as phosphoester bond. This is called as phosphoester bond. Okay. So the phosphoester bond, what we have seen, the phosphoester bond, it is formed between carbon 5 of sugar and phosphate group, right? It is formed between phosphate group and carbon 5 of sugar. The phosphoester bond is formed between carbon 5 and sugar and phosphate, while the glycosidic bond is formed between the carbon 1 of the sugar and nitrogenous base. Okay. Now, this structure that we are seeing, since it has only one phosphate, it is known as monophosphate. So the name you can also write as nucleotide monophosphate because it has only one phosphate. Okay, so it is nucleotide monophosphate. Now, if you want to draw the structure of triphosphate, because we have studied about the ATP, right, in briefly, that ATP stands for adenosine triphosphate, and it is the powerhouse of the cell. Like it produces the energy, right? So the mitochondria generate ATP, and ATP is what? It is the energy, form of energy, right? So if you want to draw the structure of ATP, ATP is also nucleotide because it is adenosine triphosphate. It has phosphate and it has nucleoside, right? So the structure of ATP, if you want to draw, so you have this thing, the sugar, right? You have this sugar, and here, because it is adenosine, you will have adenine here. Now, how we'll go about this? So we have CS2, then we have O, we have P, right? Then again, it is one phosphate. It will be then get connected with another phosphate, and then it will again get connected with the another phosphate. This is O, this is O, this is O, and it is connected with OH, OH, and OH. So we have three phosphate, right? This is 1, this is 2, and this is 3. So this is the structure of what? ATP, or adenosine triphosphate. So understood? So if we have diphosphate, we'll have 2 phosphate here. If we have triphosphate, we'll have 3 phosphate here. And if we have monophosphate, we will have only 1 phosphate attached to this 5' carbon of the sugar. Okay. So this is the example of adenosine triphosphate. Okay. So this is about the nucleotide structure. And we understood how we can draw monophosphate, diphosphate, and triphosphate. Now, let's talk about the nomenclature. Because we are saying that we have nucleoside, we have nucleotide, we have RNA, we have DNA. Then what are these bases? Or this base, like this base means nitrogenous base. This nitrogenous base. These are nitrogenous base. So what these nitrogenous bases are called when they are either in RNA, when they are in DNA, when they are in nucleotide, and when they are in nucleoside. Okay. So the nomenclature that we are here seeing is in the nucleoside. So this nomenclature is in the nucleoside, that is in the case of RNA and in the case of DNA. Okay. So the adenine in the case of ribonucleoside, or in the case of RNA, we call it adenosine. Okay. So if it is a nucleoside means it will have just adenine as a nitrogenous base plus ribose sugar, right? Plus ribose sugar. So it will be called as adenosine. The cytosine is called as cytidine. Guanine is called as guanosine, and uracil is called as uridine, because we do not have thymine in the case, right? This hypoxanthine, this thing you need not to remember for now. These are not important. Okay. Then in the case of deoxyribonucleoside, what we'll call, we add deoxy. Okay. If it's nucleoside, we'll just add deoxy. So it will become deoxyadenosine, deoxycytidine, deoxyguanosine, and deoxyuridine. So if it's a nucleotide, you will call adenine as adenosine or deoxyadenosine. If it's cytosine, you will call it cytidine or deoxycytidine, and likewise. Okay. But if it is nucleotide, if adenine is present in nucleotide, means it has also phosphate group attached to it, then we'll call it as adenylate. If it's adenine, we'll call it adenylate. If it's cytosine, we'll call it as cytidylate. If we have guanine, we'll call it guanylate. And if we have uracil, we'll call it as uridylate. And if we have thymine, we'll call it thymylate. right? We'll call it as thymidate. Okay. So it is in the case of RNA. In the case of DNA, it will become deoxyadenylate, deoxycytidylate, deoxyguanylate, and deoxyuridylate. Okay. So, sorry, in the case of DNA, we do not have uracil, right? So we'll call it deoxythymidate. Okay. Thymidylate, sorry, thymidate. Then this is about the nomenclature. The next thing that we'll study now is about the dinucleotide. Okay, so how do we form a dinucleotide? Because we see that the nucleic acid is a chain of polynucleotide, right? So before moving to the polynucleotide structure, let's see how a dinucleotide is formed. So we have seen two types of bond till now. One is the glycosidic bond that is formed between the base and the sugar, and the second is phosphoester bond that is formed between the carbon, sugar, and the phosphate. Now, what do we have in the dinucleotide? So, for the dinucleotide, it means two nucleotides. So, let's draw the first nucleotide. This is your first nucleotide, right? Suppose here it is a base, and then you have CS2, O, P, O, OH, and OH. Okay, and this is what? This is your 5' end because this is carbon, right? Then we have here OH at the 3. This is the 3. Okay, this is one nucleotide. Now let's draw the second nucleotide. The second nucleotide will be like this. Okay, the second nucleotide, suppose there is some random base, adenine, guanine, cytosine, anything. Then you have what? You have CS2, you have O, you have P, and you have OH, right? You have O and you have OH. This is your, and then you have OH. This is your 3', right? Now, how a dinucleotide is formed? So, in order to form a dinucleotide, what happens? This OH and this O, it forms a bond and it causes release of water. So, it will form a bond. It will, this OH and O will form a bond, and this will cause release of water. And the resulting structure will look like this. Okay, the resulting structure will look like this. We can see we have here the 5' end. This is your 5' end, and this is your 3' end. This is your 3' end. And the linkage that is formed, this linkage is known as phosphodiester linkage. Okay, this is known as phosphodiester linkage. And this phosphodiester linkage is formed between the 3' OH of one nucleotide. 3' OH of one nucleotide. Second, it is formed between the 3' OH of is formed between the 3' OH of one nucleotide sugar, right? That is this sugar, one nucleotide sugar, and 5' phosphate. 5' phosphate of other nucleotide. 5' phosphate of other nucleotide. 5' phosphate of other nucleotide, right? So it is forming between the 5' phosphate. This is your 5' phosphate, and this is 3' OH of one nucleotide. And this bond is known as phosphodiester linkage. Okay. So what we are seeing here, that it has one free 5' end, right? So it goes like this, because now if a next nucleotide will come, it will join here, right? And then the next will join here. So it will go like this, 5' to 3' direction. So this is the way of representing the direction of the nucleotide. Okay. Now we have understood about the dinucleotide. Now let's move to the structure of polynucleotide. Okay. So polynucleotide means we will have multiple nucleotides, right? So let's draw. So this is your first nucleotide. This will be your first nucleotide. Here a base is attached. Here you will have OH, right? Then here, and what you will have CS2 phosphate. You will have phosphate group, right? Then you will have second nucleotide. You will have second nucleotide. Again, the base will be attached here. And then here you will have CS2 phosphate, second phosphate group. And then here you will have OH. This is your 3' end. This is your 3' end. This is your 5' end. This is your 5' end, right? Then again, the third nucleotide will come. and this will be arranged the similar fashion. Here will be the base CS2P, phosphate I'm representing by P, and then you will have OH, and likewise. So this is a 3 prime, this is a 5 prime. Now, what they will do, they will link together, right? And they will link together and they will form a bond. And this bond formation will lead to the release of water molecule, right? So the bond will form between this, the bond will be formed like this. The H will be removed, right? And this bond will be formed. The bond will be formed, right? So this is how the polynucleotide will be formed. But what we are seeing here, that this polynucleotide will have free 5 prime end. And what do we have with the 5 prime end? We have phosphate group. So it will have free 5 prime end that will have phosphate group. And then it will have free 3 prime end, and that will have hydroxyl group. Right? That will have hydroxyl group. So whenever this chain terminates, it will be read like this. Here it will have 5 prime, and then it will go like this. New nucleotides will add, and the end will have 3 prime 3 hydroxyl group. So this is the direction of this polynucleotide. Okay. Now, the second thing, if we talk, how many phosphodiester bonds will be present? So in a polynucleotide, number of phosphodiester bonds. So how many number of phosphodiester bonds will be present? It will be n-1. How? Here we have 3 nucleotides, right? 1, 2, 3. And if we talk about the phosphodiester bond, how many we have? We have 1 and 2, right? So the number of phosphodiester bond will be n-1, where n is what? Number of nucleotides. So if a polynucleotide chain is of 10 nucleotides, then the phosphodiester bond will be formed as 9. Okay. So this is the structure of polynucleotide. And with this, we also understood that what is the directionality, or what is 5 prime end or 3 prime end. Okay. And why do we call it 5 prime end and 3 prime end? Now, with this information in background, let's study the structure of DNA. So DNA, we already know that it is a genetic material in most of the organism. And the structure of DNA, is given by Watson and Crick. So Watson and Crick are the two scientists who gave the structure of DNA. Now, what is DNA? So if you look at the structure, it is double helical structure. We have seen the threads, right, or wool. So the DNA, imagine the structure of DNA. If we unwrap the wool, we can see 2 different threads coming out of it, right? And they are arranged in a helical form. So the structure of DNA is double helical structure. It is double helical structure. Okay. Now, the second thing, if we talk about the directionality, so it is right-handed. The structure is right-handed, which is present in our side. So it is right-handed. Okay. Now, what I said, that it is double helical structure, right? It is double helical structure. So it consists of 2 strands. Means it consists of 2 strands, right? Double helical means it consists of 2 strands. Now, these 2 strands, these are anti-parallel to each other. So 2 strands are anti-parallel to each other. Now, what do I mean by anti-parallel? So by anti-parallel, I simply means that we can see. So this we have just studied about the directionality. So suppose this is your 5 prime end of this chain. This chain goes like this, and this is your 3 prime. This is your 3 prime end, right? So in this strand, this will be your 5 prime end, and this will be your 3 prime end. Okay. So this means one chain is running in 5 prime to 3 prime direction. In simple terms, this is your one chain, which is running from 5 prime to 3 prime direction, and the other chain will be here. It will have 3 prime end, and here it will have 5 prime end. Okay. This is what I mean by anti-parallel to each other. Okay. Now, the backbone. This is the backbone. Okay. This is the backbone. So the backbone is of DNA is made up of sugar plus phosphate. It is made up of sugar plus phosphate. And if you remember, I told that the nitrogenous base are hydrophobic in nature. So they will be inside, right? They will not be in contact with water. So the hydrophobic chain or the nitrogenous base are this one. These are the nitrogenous bases. Okay, these are the nitrogenous bases. Okay. Now, if you look at the structure, what we can see that the purple one here is representing the thymine, right? It is representing the thymine, while the green one is representing the adenine. So, if you see the structure everywhere, the adenine is bonding with the thymine, right? So, in the DNA, adenine bonds with thymine. Okay. Similarly, if you look at the cytosine and guanine, so the cytosine is the red one. Cytosine here, it is here. Cytosine is the red one, and blue one is guanine. So everywhere the cytosine is bonding with guanine. So cytosine bonds with guanine. Okay. So cytosine bonds with guanine. So this is the structure of DNA. Now, in the DNA, we have certain other things. So the first thing is, let's draw the structure again. Sorry. Okay. 1 second. Okay. So we have one more thing about the structure. So if you draw the structure like this, so this is DNA structure, right? So here, from here to here, from here till here, this is called as one turn. This is called as one turn. And one turn means 360 degree. 360 degree. So one turn of ellipse constitute 360 degree. Okay? Constitute 360 degree. Now, in one turn, the number of base pairs, the bases that I am saying adenine, guanine, thymine, it will be 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So, in each turn, the number of base pairs, by base pairs, what I mean? By base pairs, I mean this thing. Okay. By base pair, I mean this thing. This is one base pair, this is 2, this is 3, because it consists of 2 bases. The number of base pairs equals to 10. Okay? The number of base pairs equals to 10. So, if the number of base pairs in 360 degrees is 10, then at how much angle one base pair is rotating? So, what we can say that each base pair that I am representing by BP rotates, each base pair rotates 36 degrees Celsius, 36 degrees in each ascent. So, in this, each base pair is rotated by 36 degrees. Okay. Now, the length of each turn. The length of each turn is how much? 34 nanometer. Okay. The length of each turn is 34 nanometer. So, the length of what you can write, length of each base pair will be, because there are 10 base pairs in each turn, and the length of each turn is 34 base pair. The length of each base pair will be 3.4 nanometer, right? So the length will be, it's 3 point, the length of each turn is 3.4 nanometer. Okay, the length is 3.4 nanometer. The length is 3.4 nanometer. So this is 3.4. So this will become your 0.34 nanometer. So length of each base pair will become 0.34 nanometer. Now, apart from this, one important thing, the adenine, we saw in the previous slide that adenine bonds with thymine. But with what bond? What is the name of that bond? The bond is hydrogen bond. Okay. So adenine bonds with thymine. Adenine bonds with thymine by 2 hydrogen bonds. Okay, by 2 hydrogen bonds. Okay, so it will be represented by A, T. While guanine, the guanine binds with cytosine. Guanine binds with cytosine by 3 hydrogen bonds. Okay, so it will be represented by guanine cytosine. Okay, so if someone asks that we have two DNA, one DNA have high GC content, like high guanine and cytosine, while another have high adenine to thymine content, then which will be more stable? So since we know that guanine is bonding with cytosine by 3 bonds, so it will make the structure more stable, right? And hence the DNA which has high GC content, the right DNA with high GC content is more stable, okay, because it has 3 hydrogen bonds. Okay, now the last topic of this chapter, biomolecule, is Chargaff rule. So Chargaff is a scientist who gave certain rules regarding the DNA composition. So the first is amount of purine. The first rule is amount of purine equals to amount of pyrimidine. Okay, equals to amount of pyrimidine. So what it means? We have purine. We have seen what is purine. Purine is adenine plus guanine, and pyrimidine is either adenine plus thymine in the case of DNA. Sorry, not adenine, cytosine. Cytosine plus thymine in the case of DNA, or cytosine plus uracil in the case of RNA. So the amount of purine equals to amount of pyrimidine, means adenine plus guanine will always be equals to cytosine plus thymine or cytosine plus uracil. Okay. The second thing is amount of adenine, because we know adenine is pairing with thymine, right? So amount of adenine equals to amount of thymine. If it is DNA, right? And if it is RNA, it will be equal to amount of uracil, right? Now, amount of guanine. We know guanine pairs with cytosine. The amount of guanine will be equal to amount of cytosine. Okay. With this comes certain numericals. Okay. Then the third important thing is amount of sugar equals to amount of phosphate. Amount of sugar equals to amount of phosphate. That is true. You can see from the structure, right? Because the phosphate is attached to each sugar. Hence, the amount of sugar equals to amount of phosphate. And the last rule is that the ratio of A plus T, that adenine plus thymine ratio to guanine plus cytosine, is constant for a species. So if you talk about the human, so the ratio of, if you sum adenine plus thymine and divide it by, take a ratio of guanine plus cytosine, this will be constant for all the humans. Okay. So this, but it can vary. Like if you compare the dog with the human, so this ratio will vary. But among all the humans, this ratio will be same. Okay. So this is the Chargaff rule. And with the Chargaff rule comes certain numericals. For example, let's say if you have adenine 30%, then tell or what will be the component of other nucleotides in DNA. In DNA, if you have adenine 30%, then what will be the composition of thymine, guanine, and cytosine? So with Chargaff rule, we can see. So adenine is 30%, and we know that adenine is equal to thymine. So thymine will become 30%, right? The total will be 100%. Total we have 100%. Now adenine plus thymine is your 30 plus 30 is equal to 60%. So the remaining will be G plus C. G plus C will be 100 minus 60. That will be your 40. And we know guanine is equal to cytosine. So guanine and cytosine will become 20%, right? So your thymine will be 30%, guanine will be 20%, and cytosine will be 20%. So this is all about the biomolecules. We have completed all the four major biomolecules, that is your protein, carbohydrate, lipids, and nucleic acids. And with this, we are ending this chapter. That's all. Thank you.